it's pretty interesting that before my talk, there was this discussion about the cost. And our vaccine definitely going to cost you very, very little. So, because our vaccine is produced in E. coli system, it's a very standard system. And so not only can produce really rapidly, but also very economically. So VaxInnate is a vaccine company. One of the big projects is a flu vaccine. Our technology is based on the toll-like receptor, which links the TLR5 ligands flagellin with a specific disease antigen, and the resulting recombinant vaccines can dramatically improve the immunogenicity and efficacy. And we have successfully produced a flu vaccine, and both for pandemic vaccines and seasonal flu vaccines. Both vaccines have entered in clinical trials. Both vaccines proved to be safe and immunogenic. So, and today I'm gonna just focus on flu B vaccine design and development. Traditional flu vaccine has three components: one H1, one H3, one B, and more recently the quadrivalent flu vaccine was introduced into market. And in addition to H1, H3, also included two flu Bs from both lineages, the Yamagata lineage and Victoria lineage. And previously, we have developed flu A vaccine that include H1, H3, and H5. They both, all these are immunogenic and safe in preclinical animal models and also in human clinical trials. But when we apply this method into flu B vaccine design, and vaccine was found to be poor triggers of TLR5, and consequently is poorly immunogenic. So traditionally, we place epitope concentrated HA globule head and fuse the globule head with the flagellin, either in C terminal of flagellin, we call this C-term format, or replace the domain 3 of flagellin, we call it R3 format, or place in both positions, we call it R3-2X format. However, when we apply this principle, the same principle in flu B vaccine design, and the vaccine, the immunogenicity was compromised. So in our investigation, we find that the major difference between the flu A and flu B globule head is the isoelectric point of the globule head. In flu B, it has an extremely high PI, usually about 9.0, and as compared to flu A, which is in 7 and 8 range. And flagellin, on the other hand, is very acidic. It's around 5, PI is around 5. So we thought that a positive charge and flu B HA head might interact with negative charged flagellin, and a neutral pH as a result is interfering TLR5 signaling or antigen presentation. So to test this, we introduced two negative charged residues in the linking region that connect flagellin and HA head, in order to mitigate the potential charge-charge interaction. In parallel, we also generate a second structure, construct, which will move the globular head further away from the D3 domain 3, further away from the D1 domain of flagellin, which binds to TLR5. We think that that's going to improve the TLR5 signaling, as shown here. So in traditional R3 format, we fuse the globular head with flagellin, but introduce two negative charge residues in the linking region. And in the second format, we move flagellin HA head farther away and insert into the D3 domain. We call it D3 insertion format. So in animal model, indeed the two negative charge modestly improve the immunogenicity compares with no negative charge in linking region, but the D3 insertion is the one that provide the most significant improvements in measured by HAI titer. So in order to test the flagellin function in the fusion protein, we use the in vivo cytokine assay to assess the TLR5 activity. So we measure, so basically the group of 5 boxy mice were immunized intramuscularly without vaccine, and so invested in 3 hours later, and the cytokine level, IL-6 and TNF, was measured using the CBA assay. So as shown here, in IL-6 and TNF alpha level, the negative charge modestly improved the cytokine secretion, but D3 insertion format provide the most significant improvements in cytokine level, especially IL-6 level. So in order to rule out the difference, the folding of different format may affect the antigen presentation, we use the neutralization inhibition assay, NIA assay, to assess the integrity of the HA head in the fusion protein. So NIA basically measure the ability of the vaccine candidate to bind and deplete the neutralizing antibodies presented in the immune serum, thereby to assess the integrity of neutralization epitopes in the fusion vaccines. So the higher the potentiality of the vaccine candidates, the greater depletion of the neutralizing antibodies that allows test virus to infect MDCK cells. So as shown here, so we compared R3 and D3 insertion format. Basically, they're very, very similar. So R3 format and D3 insertion format competed equally well for binding to the neutralization antibodies in the immune serum, indicating that antigenicity of the HA head is unaltered across a different format. And so the poor immunogenicity of R3 format of fljB vaccine most likely related to the TLR5 signaling. So in order to find the best insertion point, we constructed a numerous construct that has insertion points in both D3 domain and D2 domain, also included one in D1 domain. All the insertion sites are in the looped region. Theoretically, they wouldn't affect, they would allow HA head to fold independently. Also, they wouldn't affect the flagellin folding. So we screen the vaccine variants by in vivo TRR assay, also by an NIA assay. As I mentioned before, NIA is not very discriminative because they both folds independently, folds well. But the cytokine assay actually give you the good indication which one behaves the best. So in our screening, we find the two D3 insertion variants outperforms D2 insertion variants. As shown here is in vivo TRR5 assay for the representative vaccines. This is 2 D3 insertion. This is D2 insertion. You can see D3 insertion outperform D2 insertion variants. In the mouse immunogenicity studies, again, D3 insertion outperforms D2 insertion. So in order to test whether this format is generally applicable to to other flu-B lineages, we construct 2 additional kind of vaccines using Yamagata lineage, which is B forward to 2006, and currently circulating Victoria lineage, which is B Bangladesh, 2009. So as shown here, again, D3 format outperformed the R3 format, and it's elicited very robust HAI titers. And this is Victoria lineage is comparable to the commercial TIV vaccines. So finally, we test the immunogenicity and efficacy in using the historical flu-B strains, which is B Sichuan 99. And surely you see the robust immune responses measured by HII titer. And in the mouse challenge study, a group of 10 bouncy mice were immunized subcutaneously with the 0.05, 0.5 microgram of vaccine candidates, and formulation buffer as a control, immunizing day 0 and day 21 or day 42. Mice were challenged with the 5 LD50 of the B Sichuan virus. So as shown here, even in 0.5 micrograms and dose, and mice is 100% protected, and only results less than 12% weight loss. And in placebo groups, most mice succumb to the virus challenge and resolved more than 75% weight loss. So in this challenge, in this challenge study, the results shows generally support the general suitability of the D3 insertion format for flu B vaccines. So recently, the Young et al. Published the flagellin TR5 structure in Science. This structure confirmed that all known the agonism activate TRR structure form a similar dimer organization, which brings the C-terminal regions into juxtaposition so that the intracellular TR domain can initiate the signal cascade. And when flagellin binds to the TRR, the D1 domain of the flagellin is the major domain that interacts with TRR5. In this binding process, D1 domain, D2 domain actually undergoes a huge conformation change and bring the D3 domain in a different position. So because it involves a huge conformational change, and R3 format may have some sterile constraints. When we further remove our antigens in D3 domain, which is far away from the TR5 binding domain of D1, and that may release some of the constraints and provide the most rooms for to accommodate inserted antigens. So, in summary, we have developed the alternative vaccine format for influenza B vaccines, for which the HA had inserted it into domain 3 of flagellin, and we have confirmed the optimum insertion point within domain 3. We also shown that the redesigned format maximizes TRR5 signaling and immunogenicity for multiple flu B strains. And similar to A strain vaccines, the flu B vaccines can economically and efficiently produce in our standard prokaryotic system, which allow time and cost efficient production of the multivalent seasonal products, comprising both A and B strains. I want to thank our molecular biology groups and process and development group, analytical group, biology and immunology group in VaxInnate for this work. We also like to thank Dr. Andrew Ward and Ian Wisdom for the collaboration. The project was funded in part by BARDA under the Departments of Health and Human Services and VaxInnate Corporation. And thank you for your attention. Yes?
So I just wanted to ask a question about the potency of your vaccine, because I noticed that you had to give the mice half a microgram twice, and you still were getting clinical disease in the mice. They were losing some weight and recovering, whereas if we're using a recombinant hemagglutinin or inactivated hemagglutinin, we can get complete protection with generally 10 nanograms of antigen given twice, and we see no weight loss, challenged with, say, 20 LD50 compared to, I think, the 5 LD50. So that would seem to imply to me that this construct isn't terribly immunogenic or protective. Do you want to comment on that, or is it a feature of the model, or why do you need to use so much antigen?
Well, it's a subunit vaccine. It's only a domain, not even a whole HA, right? So the number of epitope presented here is just less than the whole virus. But my argument is the epitope is very concentrated and basically direct the whole, our vaccine basically direct the whole immune system to this we know it contains the protective epitopes here. So it will do the same work. For immunogenicity, it varies by antigen and antigen. In flu A case, this is a flu B vaccine probably is among the lowest immune responses, but our flu A vaccine actually at least is a very, very strong HII titers. And dose is very, very small here, and in part has to do with TR5 signaling because that basically is built-in adjuvants, right? So to us, we see this as a very strong immune responses, and even our clinical dose is very small.
But do you think that the head of the Influenza B on your construct is perfectly folded and identical to the native head? So one of the reasons for that lower immunogenicity could still be that in some way your construct is resulting in very small conformational changes in the head region, which then induces a lower quality antibody response. So without a crystal structure, I guess, of that head, which I presume you're trying to get.
I hear what you say. Actually, we got tons of questions about the folding and refolding issue. We don't have a crystal structure for B, but we do have the crystal structure of A. Actually, in Wilson's lab, published paper. So in our fusion protein with component 07, the globular head is identical to the crystal structure of the virus. And in a B, we routinely use monoclonal antibody to assess the epitopes, and it's comparable to either HA, a protein full length HA0, or well, mostly it's HA0. The response is almost identical. We have our model systems, for example, like PRA, which the epitope is very well mapped. And then we have monoclonal antibodies, and just compare the four representative regions. There is just
So in that case, if you think it's identical, the problem is you're not generating broadly neutralizing antibodies to the stem or to the cleavage domain, and that would explain it's not as protective, I think, than the current vaccine. So I'm still wondering if it was a pandemic, would I pick this vaccine? The answer is no. I would want a vaccine where I could get as many epitopes and as broad a protection as possible. But just from the data you've presented, that's what it seems to me.
So get back to the original question: is you want a variety, or you want highly concentrated or is highly effective vaccines? So there's a pros and cons, and I agree with you. But the biggest advantage here is we can respond to pandemics really fast, within weeks, and we can produce high quantity of doses in a short period of time. And none of these current vaccines can do that. So, and on top of that, and we did kind of a combination type of studies, for For animals primed with the commercial vaccines, our vaccines can boost well. In using our vaccines to prime, and commercial vaccines can also boost well. So if there's a global strategy with quick response and high efficiency, we can combine all this. So there's a position. There's a place for this type of vaccine.
I'm not saying
So.
this.
There is one question.
Yes.
The last thing that you thank you for great lecture. About if in place I am afraid of different type of vaccine, as this is very effective, I can give this and give the other that cover another serotypes, no problem?
No. For flu, we basically has all different kind of. We test H1, H2, H3, H7, H9, and all the Bs. Our company also have other programs, including C. diff, dengue. So there's a wide application for all different kind of disease target. As long as you know the protective antigen, you can engineer your protective antigen with flagellin, and hopefully give you immune protection you are looking for. So.
You mentioned
Yes.
L6 in
Uh-huh.
and I wonder whether you have looked at Th1 cytokines in your experiments. I wonder, you've measured L6 and the TNF-alpha production. I wonder you have looked at the Th1 cytokines, whether your vaccine can induce Th1 response.
Okay, it's a good question, but well, personally, I didn't do that experiments. But in the kids, I guess this is all antibody responses. It has a whole panel of cytokines, and only IL-6 and TNF give kind of indicative levels. And the rest, either in a baseline level or is saturated very, very quickly, and it's not very helpful, not very indicative. We don't. There might be timing issues and those. We haven't exhausted our cytokine assays. So. Yes.
Have you tried it nasally rather than
we did.
Because then you wouldn't get as much reactogenicity, which again is your limiting factor because of the TLR activation. So that's always going to be your I guess, stop you going up in dose. But nasally, how does it work?
We did observe a very robust immune responses, but not as beautiful as I am. So.
Okay.
Okay, thank you.